Campus in Northeast PA. I studied agricultural science and mushroom science and technology at Penn State University. I just graduated last year, and since then I've been working in a fungal biology lab, working on Fusarium, and that's what my talk is going to be on today. My passion for mycology stems from the love for the outdoors, during COVID, I learned about mycology, learned about foraging, how we can interact with nature through that way, and I really delved deep into it. I loved taking stuff home and culturing it on Petri plates in my room. That's where my passion stems from. It also stems from my experience as a cancer survivor and learning how fungi and their products may have immunomodulatory properties and also uses in conjunction with conventional chemotherapy. So let's get into it. So my talk, like I said, is on Fusarium. It's a controversial genus of fungi. And as we'll see, there's a couple controversies I'll talk about. As you may have heard, there was some national news concerning Fusarium. And then there's also some controversies within the scientific community, battles over classification. So this will be a short introductory talk. Like I said, short. If you guys, I invite you to raise your hands, ask questions. We can stretch this out if you like. So here's a bit of that international controversy I mentioned. You guys might have seen these images on the news. So some international students were caught entering the country, smuggling this fungus within their bags. And so it kind of drew up a lot of controversy and news because the fungus that they were smuggling is called Fusarium graminearum. And this fungus affects our crops, like wheat, rice, corn, all the cereal grains will get infected with this. So it causes a head blight of the crops so that they cannot grow fully and mature. Along with that, they produce metabolites that cause, as you can see, vomiting, liver damage, reproductive defects. So a lot of harmful products from these fungi. You know, as we like to romanticize fungi and say, oh, they're our friends. They like to help us out and help us connect with nature. And some are opportunistic pathogens, and you know, life finds a way, and this is their way sometimes. So as you can see, there was a lot of controversy, but then as the experts kind of examined the situation, they said, okay, there might not be too much cause for alarm here. I know my professor got a call from the Associated Press, and he gave a comment. So it is concerning that they were trying to smuggle it. They were, trying to deceive us and not do it the right way, but it was not, like a bioterrorism act. It was just, trying to get some research done the wrong way. So another controversial topic in science, as you guys may know, is taxonomy and phylogeny. So taxonomy is the classification of life into orders and domains. So there's the domain of fungi, animalia, plantae, all that. And so phylogeny is this emerging system where we have genetics technology now. Genetics emerged in the last century, and it's kind of changed the way we classify organisms. So as you can see, taxonomy kind of goes and narrows down into the genus and species level and gets very specific. Phylogeny is more the study of evolution. So through genetics, we can kind of make the tree of life and see where things branched off and develop certain characteristics. So it's very, kid-friendly here with these big, broad groups. But as we get more into the fungi, we can see, oh, this is where it branched off and produced this toxin, or this is where it branched off and was able to become a disease to certain plants. And that allows us to make inferences on things that we don't know about undiscovered fungi. And this can be a very controversial topic for scientists. We all create these names, and there may be some attachment to them, and a lot of differing opinions come up. Again, taxonomy and phylogeny. So here we have the phylogenetic tree of fungi, starting with Dicaria. And so Ascomycota and Basidiomycota split off. Basidiomycota at the bottom, those are our favorite fungi. We like to eat the mushrooms. On the bottom there's Agaricus bisporus, the white button. And so up top, Ascomycota is that group, and so Pazizomycotina, that's a really interesting group within Ascomycota. So we have Bericiomycetes, Sodariomycetes, all those mycetes. Sodariomycetes is the group that Fusarium is within. But also within that, so you can see the first image, that's of the head blight of wheat in Fusarium. Also within Pasisa mycotina are morels. That's why we gathered here. Yeah, very interesting ascomycetes. We have penicillium in this group as well, a very important medicinal fungi for antibiotics. We have truffles. Those are in Morchulaceae, some of them. A lot of or most of the lichenized fungi are also within Pasisa mycotina. And then below that, we can see where our yeasts split off. So I have a little beer icon, but that's our bread yeasts and our alcohol-producing yeasts. And then below we have Tephrinia mycotina. This one's a little more niche and microscopic, but you can find fruiting bodies in the wild of a certain club fungus that people will mistake for cordyceps. But that's probably where you'll interact with Tephrinia mycotina. So this, I just wanted to demonstrate learning all of these facts and how these things connect. This was all possible through phylogenetics. Yeah, and that's kind of influenced taxonomy. Okay, so here I'll talk about the distribution of Fusarium. So like I mentioned, it's a very important plant pathogen, but it's also a very important animal pathogen and human pathogen. So Fusarium, the majority of the species, they'll spend their time in the soil as saprotrophs. So they'll eat dead, decaying matter, kind of chill out in the soil. But then as they come across, and morels and other associated fungi do this as well, but they'll come across a host that's suitable for them. And so they're opportunistic. They find that opportunity to become a pathogen, a disease of that plant, and then they'll infect and take over that plant and cause certain defects like head blight or nodules on the stems. So like I said, it's a plant pathogen, very important animal pathogen. On the plant topic, you can see on the top right, they're pawpaw isolates. So if you know the pawpaw, that's a native fruit to us. And it's pretty niche that we have those isolates in the lab. It's an animal pathogen. Right here we have a sea turtle nest. That was one of the projects I was helping out on, organizing culture and DNA analyze the sea turtle nest. So there was a sea turtle nest. It most likely did not hatch. There were some problems with it. So scientists will go there, see that problem, and say, what happened? They think, oh, it could be a soil microbe that caused this. So they identify that, and then they send it to our lab, those samples. And then we will do genetic analysis and find out the big picture. So it can be a prominent human pathogen as well. And this is very important. So as you can see, we kind of have a hospital set up. And that's because Fusarium and other things like this can become nosocomial pathogens, which means they're transmitted through the hospital system, a sterile environment that can become unsterile through contaminations, like through a tap water system. So that's one project that we may be working on, examining how Fusarium can spread through tap water in people's showers. So the Rose project is placing a plate in the shower, the water droplets come through and drop on the plate, and then whatever's on that plate, we can infer that it was in the water system from those people's homes, and then see how that might be getting into hospitals. Okay, so a current controversy. This one was my main project. I worked at the Fusarium Research Center, along with washing plates and washing flasks and prepping plates. So Fusarium proliferatum. It's just a species. It was described and cultured as Cephalosporium by Matsushima and later reorganized as proliferatum by Gerlach at Nuremberg. And so currently a recent publication has come out trying to reclassify or reorder proliferatum as Fusarium annulatum. And on the right here I have some photos of annulatum from that paper. And as you can see, it's named after its annulate macroconidia that kind of make a C or a crescent. And just remember that shape because it'll come up later. And so the morphology of these reproductive structures at the microscopic level were used to back up their claim that Fusarium proliferatum is actually annulatum. So my project is to kind of examine their claim and go back to basics and kind of see, okay, is that the truth? So to do that, we need to know the reproductive structures of fungi. If they're going to use their claims, or these as their claims to support, we got to do that too. So I'll give a brief intro to those. We have the meiotic and sexual stages. Those are our fruiting bodies. Those are our fun to find fruiting bodies. So we have the ascocarp. That is actually the cup-like surface on the morel. So morels are actually similar to what's called a discomycete, which is kind of like an open cup fungi. And you can kind of think of all those ridges on the morel as separate cups. And so that's where the spores are produced within an ascus. And then on our lovely mushrooms, we have the basidiocarp, which are our gills or pores or whatever you have there. And then for the microscopic structures, we have the mitotic or asexual reproduction. So this is how a fungi will spread within its area that it is, for the most part. And so they'll produce conidia on conidiophores and conidia on phyllodes as well. So Fusarium proliferatum has some really interesting structures. They're called polyphyllodes. So they have multiple openings and they're also built upon each other. So as you can see on the drawing on the top right, they have really interesting, almost like antler-like protrusions. So the microconidia, those are the almond-shaped spores on the top right. Those are produced from those polyphyllodes. And macroconidia are produced on Sporodokia. We're not too interested in the Sporodokia for this identification because we have this drawing. So usually when you start a culture, you start what's called, or you preserve what's called a holotype. That is the genetic precursor for that culture, for that species. So unfortunately, this holotype was created in Japan by Matsushima, and it was lost in one of their natural disaster earthquakes. I'm not sure which one, but so at some point this culture was lost, and so we need to go back and try to redo it basically. So my job was looking at this drawing and going back to the microscope and the slide and seeing where we can find those structures in this fungi. So, and as you can see, so Annulatum had those really crescent-shaped macroconidia, and this is a macroconidia from proliferatum. They're a little more open. And so this is how we do that. This is how we get that evidence. There's my little battle station up top. I took a little panoramic. We have a dissecting scope, and then a differential interference contrast scope, and then a computer setting up, taking that information. So the middle microscope has a Nikon camera that's reading what I see on the slide, and then we pull out a bar and it can switch between. So they were grown to get the cultures to produce the structures that we want. They were grown on specific media. It's SNA media. I forget what it stands for. It's like some long German name or something. But so, and they were given filter paper to grow off of. So these structures like to grow off of the plants that they infect. And to kind of emulate that, we put a sterile filter paper on the slide, and they can kind of produce their aerial hyphae and reproductive structures for there. And so, yeah, a lot of the work was done with Nikon software and a Nikon camera, but also, physically moving things on the microscope. And so I have some photos that I took on the right here of these polyphyllodes, and you can kind of see how they branch and connect to each other. And there's some microconidia scattered throughout the images. So, but these are not cleaned. These haven't been photoshopped or edited. As you can see in the papers, they line them up all nice. And then, yeah, in the middle you can see a little mix of macroconidia and microconidia. So, and we're not really seeing those hard crescent shape that you would assume to see in Annulatum. So that project's still ongoing. Still working on it. Getting the fungus to cooperate a lot of times is tough. It's a waiting game, gauging when these structures are produced, and then conditions in the lab are always changing with the weather. We try to keep it a climate controlled, but it's an old building from the 1920s, so a little bit established. So looking forward, I just wanted to kind of talk about the state of academia and why we're here. So Penn State, it's a great place to get your start, but as I've learned that at least in the mushroom production side, they're kind of lagging behind industry. So it's a good place to start and get your basic education, but I want to emphasize that you can be self-taught. You can be a so-called amateur and learn these things and spread it to other people like so many people here do. So yeah, the current state of academia, it's really hard to get a master's program right now, especially in these more niche areas of study. Funding has been cut pretty extensively. So, they offer you, you could fund it yourself and take loans, and it's not looking too appealing. But so industry might be a way to go for people. We did just have a visit from a chief innovative officer of a company come into the university and talk to students about how to prepare for a role in the industry because of the changing academic landscape. And so what can we do as a community? I think it's great that we have events like this. We have people from both academia and, you know, the amateur side of things coming, converging, sharing what they know, spreading their knowledge, teaching people. So yeah, I appreciate y'all coming out here and supporting people. It's good to see that events like this are going on. And yeah. Is there any questions? I could go back on the slides, rehash some things, but yeah, any comments, questions?
What happened with the turtle eggs?
The turtle eggs, those are, it's still up for, you know, there's no conclusion to that. There hasn't been a project on it, but, you know, we'll get samples. We'll get a snake or eggs or something sent to us in the mail. Sometimes it's samples, sometimes it's Petri plates. I prefer the Petri plates. But yeah, so that could be a future project. But right now, our job is basically just getting the samples, examining them like through genetic analysis. So we'll get a short read and a long read. So a short read is just one sequence of the whole genome, and then a long read is the whole genome. An interesting thing about Fusarium is that their genomes are quite large. So a Pleurotus, an oyster mushroom, it's about 32 megabase pairs, I believe. And then a Fusarium genus can be twice the size of that, which is kind of crazy because you have all this genetic information for this little fungus, but then we realize, oh, there's all these accessory genes for pathogenicity, where it gets that opportunity and those genes activate. So yeah, it's really interesting. Yeah, genetics has kind of opened the door to understand. Yes, Lou.
For people who are like in a set career path or doing corporate work and haven't had an academic exposure to science, like how would you encourage them, or are there strategies to get involved at the university or college level that are like more friendly to amateurs?
Yes, that's interesting. Because yeah, most college clubs will require that you're attending the college. But yeah, I would look for more outreach events like this, and yeah, that's something I kind of want to see from the college too, is more interaction with events like this and local clubs. So yeah, we're hoping to see that.
Woo!